first of all, this could be a true and false question. Fungal infections are harder to treat than bacterial infections, true or false, in general? True, because they're eukaryotic cells. So what is the big, that could be a question right here. What's the biggest target in the fungi for antifungals? It's exactly, you think it's the cell wall because we don't have a cell wall, they do, so we should target it. The cell wall is not. It's actually the cell membrane, specifically which sterol? Ergosterol. Okay. You have some that disrupt it, you have some that inhibit it. Okay. We talked about amphotericin B, terbinafine, the azoles. Then you have nucleic acid inhibitors, 5-fluorocytosine. So again, there's probably about 30 total antibiotics. You're going to get about 9 of them to factor. I mean, 9 into questions. So, any questions about antibiotics? Now I'm going to spend a little bit more time on the old because it's really old, I guess. The new material is, honestly, it's not complicated, it's just a lot of definitions, etc. But we'll try to target everything. Okay, so then we come into exam 3 material, which is the freshest. So that should be, and then the class, I think that was, most of you did best on exam 3. But here you have 3 chapters, you have 7, you have 8, and you have 9, because 10, regulation of gene expression, is not undefined. And with 7, it's DNA and RNA structure and replication. 8 is transcription translation. 9 is microbial genetics. So we'll start with 7. And remember, that's the only reading material you have to know. So what is that reading material? This is stuff that honestly you should, as a biology or BMS or any bio-related, I mean, you should know. This is standard nucleic acid chemistry. So for example, what are the bases in DNA? What are the bases in RNA? If you are given the percentage of one base, can you calculate the rest? Right? So for example, if you have a bacterial, and be careful what the question is asking, right? So if you are told you have a nucleic acid that has 20% adenine, you have a nucleic acid molecule that is 20% adenine, what is the percent of cytosine? See, many of you are rushing into, you have no idea. It's not 30. Why is it 30? Because I just said nucleic acid. The only way you can know it's 30 is if what? If it's double-stranded. The only way you can tell the rest if it's double-stranded. If it's single-stranded, you have no idea. Because it could be anything, really. So if you're told that it's double-stranded DNA, and you're told that adenine is 20, then the rest can be easily figured out. It could be viral DNA, which has, right, which could be single-stranded. So, alternatively, you can be asked something like this. So these are the bases. This is the percentage. And this is the base. It's probably easier to read. Okay, so A is 20, let's say. C is, I don't know, 30, let's say. G is 30. T is 0. And U is what's left, 20. And then here are our choices real quick. Single-stranded DNA, single-stranded RNA, double-stranded DNA, double-stranded RNA. Which one is the most likely? Okay, so first of all, you look at it. There's 0 T, there's a bunch of U. It's going to be RNA. So you automatically rule out DNA. That doesn't make sense. So now the next step, is it single or double-stranded? And the answer, it should be double-stranded, at least most likely. Why? Because if it's double-stranded, the A would be equal to the U, which it is. The C is equal to the G. So it's most likely double-stranded. If any one of these is not equal, it's got to be single-stranded. You have to also know base complementarity, right? So if I give you one sequence and ask you to determine the complementary sequence, you look for A's bonding to T's or U's, C's bonding to G's, but they also have to be what? Because many of you did this mistake on the exam, or there's only two rules of nucleic acid chemistry that are important here. When you have binding of DNA and RNA to each other, they have to bind, first of all, complementarily, meaning A to TU, C to G, and the next one they have to bind anti-parallel manner. So you can't have a 5' binding a 5', right? So they have to be 5' to 3'. They have to be anti-parallel. So make sure you remember that. Okay? But these are the big ones. What does anti-parallel mean? One goes 5 to 3, one goes 3 to 5. What are the purines, adenines, and guanines? What are the pyrimidines, cytosine, uracil, thymine? Which is bigger, adenine or cytosine? Which is bigger, adenine or cytosine? Adenine. Why? Because adenine is a purine, so it has 2 rings. Pyrimidine is 1 ring, right? So this would be a good segue into replication. True or false? Origins of replication should be rich with G's and C's over A's and T's. Origins of replication. That would be false. Why? Because G's and T's are more stable because they're triple bonded. A's and T's are weaker to open up. So if you have an origin which needs to open up, it makes sense to have it as a rich in A's and T's. Okay, so replication, still chapter 7. You will get several questions on polymerases. Right? So please know the polymerases in bacteria, what they do, the polymerases in eukaryotes, what they do, right? So real quick, in prokaryotes, you have many of them, just know the big 5. Alpha, beta, gamma, delta, epsilon. Alpha, delta are DNA of the nucleus. Gamma is mitochondrial, it's easy. Gamma, there's an M, mitochondria. You can come up with a better mnemonic if you want. And the others, beta and epsilon, are repair. Okay? With bacteria, you have 3 big ones, 1, 2, and 3. The main enzyme, the big work enzyme, is DNA pole 3. Pole 1, small but important role, it removes the primers. Pole 2, mostly repair. The origin is called OREC. You have to know the proteins in order. Right? So you will get a question, these are the proteins, put them in order. So let me give you an example, and I can use it to summarize replication. In fact, yeah, summarize replication.